was just thinking I was going to ask her, so.
It's a good guess.
We have about 30 meters to go on transect.
No, I mean the percentage of the whole track.
Oh,
for
this
the
is
day.
the second waypoint out of this. Well,
Out of how many? Are
Bob,
they
you're gonna.
evenly spaced?
Nope. Sorry, somebody in the back row had a quick answer, maybe.
Okay.
How far? We did have a 1,400 meter
Yeah.
track, but we're not going to be able to make that.
Let me.
And I don't want to, I was thinking about coring, but Bob, you don't want to start coring now because you're going to
Well,
go off watch
we're on a
very
transit.
shortly.
We're on a transit here.
I know.
I mean, I could go, Jess knows how to do cores. I mean, it's not a problem there. But I could also go eat and come back. But I want to go talk to Dwight and see what's going on.
We need to know a little bit about whether we're pulling at 345 or not.
We need to talk about it. So I don't want to do it over the SPL in here.
So that, I know you hate this and can't see it, but the end of the dive is there. And we've covered about that much.
Yeah.
Okay.
Yeah.
Bridge
We're just
nav,
going to
another
get
step,
into
same
this
bearing.
steep stuff and when we go off watch.
Yeah, have to watch.
Yes, correct. Thank you.
Is that a little skate? What is that thing? Yeah, I think it's a little tiny skate.
Very nice.
Cool.
It's a baby.
Oh, nice Argus too. Really clear.
So I don't want to stay zoomed in on Argus all the time because we're not seeing what's happening with the tether and.
Makes me want to.
You're so close to
Makes
the
me
end of
think my
transect,
delta
though.
0.
Yeah, I mean, it's cool to zoom in once in a while, but I don't want to fly around like that all the time.
Got it.
We're not seeing what we're doing.
Some nice rocks there. You're very close to the end of it. Another 10 meters or so. So since we are approaching end of transect, do you want to explore at the end or you want to keep moving towards the waypoint?
We keep moving towards waypoint.
Okay.
Or do you want to blast out tube cores?
Well, I'm just wondering, it's 10 minutes to 12. Are you about to go?
Well,
I don't want to start something.
no?
I think we'll just keep going.
Okay, roger that.
Pretty sure there'll be a sedimented area at the end of the
Look at all these rocks.
That's a cool
steep
rock.
spot.
Shiny.
What do those look like?
Those look like crusts. Yeah, ferromanganese coated basalts. And
Wow.
Are you there yet?
Pretty much.
It's
One
the
more
end?
meter just for good luck. All right, let's call that end of transect.
Okay, but we're going to keep going?
That's the
Or
word
do
I
you want
hear.
to look around here? You want to rock? Or?
No, keep going.
Keep going.
Okay.
Oh, I can't. Can you read me? Let's see it. 038766.
Yep. And I would say we're off transect, so you guys can do whatever you want with the camera. We might start seeing some increase in slope, and it looks like a lot of rocks coming up. Mezzo looks pretty bumpy.
like a big debris field.
Hey, Al, can we zoom in on that? Okay.
Paul, do you want Faria or whatever this sponge is?
A lot of cool biology.
Want some of this? I think it's a Faria sponge. You think it's a bryozoan?
Are we considering a sample? I can hold position.
Yes, we are considering a sample. We're changing the watch here, so.
Okay, I'm going to have the ship hold
Yeah.
position then, if that's okay.
Yeah.
Bridge nav, hold position.
It's a cnidarian of some sort. I don't know. It's not. It could be an emballula, but I don't think it is. Something else.
We got more zoom there now?
You're right. It does look like a bryozoan up close. So you want some?
Did you get an image capture here?
So you want some of this?
Good capture.
Thank you. Are we going to sample here?
Yes. We need to
What are we
tell them.
sampling?
We'd like to sample this bryozoan, the white thing that looks a little bit like a sponge.
Okay. Can you zoom out? And how are we doing that? Is that going to be a slurp?
You okay with slurping it, Paul? You okay with slurping it? Sure, that would
Okay.
work. We want one of the coarse boxes, so 4, 5, 6, or 7.
Okay.
The whole big thing? Okay, he's asked for the whole thing.
Okay. Where's he going to live? Is he going to...
In the slurp.
Oh, slurp the whole thing. Okay.
It's really fragile and it'll break in pieces.
Okay.
Hopefully not clogging the slurp.
Yeah, I don't think that... I doubt that would clog the slurp.
Is that a hydroid? Oh, okay. I knew it was an idarian, but do you want that too?
Nobody
Okay.
logged that. We need a data logger to log that. We need a data logger to log the solitary hydroid.
None of us know how to do it.
Which slurp
Well,
jar
they're
did
right
you want?
there.
They're not supposed to leave until the next watch comes. I don't know.
Which slurp jar did you want? 3? Sorry.
They're right.
Good.
All right. Chill for a moment.
The logs up.
I'll just get the grip ready.
What number is it on now?
It's on 3,
Okay.
but I don't know which one they want, so we're not.
There's a, there you go, they're doing our scuttlebutt.
Do you remember which one's the
One-
course?
1, 2, and 3 are the smaller mesh.
Oh, so we want 4.
4, 5, yeah, the higher numbers are the regular.
Give me another spin on the
sample
wheel
jar.
then. Yeah, that's perfect.
Okay. How much beans do you want?
No beans yet.
Okay.
We're gonna get ready and then we're gonna pass the beans off to the next watch.
Okay.
What's on the end of that? What is that?
Kind of looks like tape.
Why is there tape on it?
I don't know.
Can we zoom in on that?
Okay.
What the?
Oh no. I
Got some microplastics in the sample there. Okay, zoom out. I guess we can just do it. We're already here.
Yeah.
Well, I guess we need some confirmation on jar 4 being okay.
Absolutely, taking a look at that. Jar 4 is open.
And that's the coarse mesh.
It is coarse mesh, yes.
So this is what we wanted. So zoom in. All right, Kylie, give it
Beans.
some beans.
100%.
Getting some good screen grabs.
You can.
Am I drifting off?
No, you must said you have a good view in bubble of the depth.
Can you zoom out a little bit? Huh. That's a tougher sponge than it first appeared. Yeah. Guess I gotta wipe it on the rock there or something. Yeah, Mr. Crab.
That parallel crab wants to bite of that.
Okay. Hey, everyone. The second Science Watch is taking over for the second half of this dive.
There you go.
And we're
As it's being sampled? Okay.
continuing with
Right,
this
right.
Bryozoan collection.
This is a Faria sponge.
Anything going in the jar yet?
Looks a little large.
Pieces of it.
Well, that sucked it up. We want the
rest
whole,
of it.
the bits.
Oh, now it's in the, yeah, it's in there now.
Beautiful.
Do we want the rest of it, or are we gonna leave it?
They did initially want the whole thing.
I thought they did want the whole thing.
They did want the whole thing.
Okay. Can you zoom
Yes,
back
yes,
in?
please. The whole thing if possible.
Fantastic.
Yeah, correction. This is a sponge, not a Bryozoan.
You got full beans on it?
Yeah, it's 100%.
Oh, come back here.
Is it going?
This is a Faria sponge, and it's sample ID number NA124 038.
There's
And
something orange living in it?
And it's going in slurp jar number 4.
Looks like there's some little lobsters living in here, in the sponge. Orange.
038, was that the right number?
Yes, 038.
There's usually all sorts of little fauna inside too. There's ophiuroids sometimes, squat lobsters.
Yep. Scale worms.
Polynodes, yeah.
My favorites.
Yep.
Yeah. But the lobster looks kind of unusual.
Someone was
Yeah.
hiding out in there.
Well, it looks like we got that one.
Awesome.
Cool.
Good job.
These Hercules sponges come in a lot of different shapes and sizes. Branched, these are the bushy types. We actually came upon a Hercules reef in our last
Can we
dive.
zoom out? Or is that zoomed out?
That's zoomed out.
Last dive on, where were we?
Sorry, Bob, before you
Footprint.
stow, could you try and get the little lobster that's just to the left on the rock, just in front of you? That was in the sponges, yeah.
Are we sucking still?
That's
Yeah.
it, yeah. Thank you.
And before you put the suction away, I think we're
Yep.
going to try something a little different. We're going to try to collect a sediment sample with the suction, since we
So-
haven't had a chance to do any cores. So
Okay.
I think we do this.
Okay, well, I'm going to actually pass the watch on.
Okay.
Just can pick up and do that.
Okay, sounds good.
I'll turn
Quick.
off suction for you for right now.
Copy.
Did that second piece of the sponge go through? I might have missed that.
It looks to be in there.
I saw all of it
fantastic.
get in, yeah.
You can
Okay.
see it in the lower right display in the mid-center of the screen.
Awesome. Thank you.
Yeah.
Okay, so we changed out the science team, and we're also changing out the pilots. So while we take a moment break, I'll just introduce myself. My name is Paul Jensen. I'm at Scripps Oceanography. I'm a microbiologist. I don't know if the other team members want to introduce themselves. Greg?
Yeah. Hi, it's Greg Rouse here, also from Scripps. And I work on animal biodiversity, so I'll be helping identify the various animals we collect today, and that'll inform the ecologists and Paul in the pharmaceutical side of things for vouchering specimens into the collection at Scripps for future use.
Hi, I'm Caitlin Kramer. I'm a graduate student at Scripps Institution of Oceanography. I'm a student in Paul Jensen's lab, so I'm also a microbiologist, really interested in all of the different microbes that are living in all the environments that we'll hopefully be looking at today in both the sediments and living as symbionts or on top of the creatures and rocks that we'll be sampling.
Hey, folks.
And I am Kyla Pearson. I am also a graduate student at Scripps Institution of Oceanography. Lucky me, lucky us. Being out here on the Nautilus, it's just absolutely a dream come true. It's phenomenal, and I'm honored to be out here. And I am in Dr. Greg Rouse's lab, and I study deep sea worms.
All right. So that's the introductions for the second Science Watch. And if you're just joining us, we're on the San Juan Seamound, about 150 miles off the west of the coast of California. And we're thinking about using the suction to collect a sediment sample, since we haven't had a chance to do any push cores. And it's not the usual way we do this, but we're thinking if we use the coarse filter, which would be either 4, 5, or
Sorry.
either, but okay, yeah, 5. If we could suction into 5, and I think our goal is to see if we can't get at least a couple centimeters deep of sediment into that bin.
Number 5, roger that. You want to go to like maybe 30%?
Hey, I don't know, Data, if you want to make a note that that lobster that we wanted to suck up into the last jar made it into the flush jar.
Oh, it made it into the flush jar.
So it did
Absolutely.
it did not go into the last jar.
Okay, absolutely.
The flush holes may be big enough that it will exit, so it may not remain there.
Okay,
But
sounds good.
Thanks for the info.
yeah.
All right, go ahead and line up on number 5 there, Jake.
They seem a bit floaty, so maybe you want to go to maybe 50% suction instead of 90.
And we had a nice ID from Mary Wixton in the science chat saying that little orange crustacean in the sponge might have been a Echinaxius baja,
So
that
Greg,
lobster.
sorry, so for this sample, you would like the top kind of rows and anywhere around here in the sediment area
Yeah.
be fine?
Yeah, I think we don't want to go too deep down, so we wanna suction up sort of the top couple of centimeters.
Okay, sure thing. And just to confirm, slurp number 5.
Slurp number 5, yes please.
Roger that. All right, Jake, go ahead.
Roger, 50%.
Raj.
Shall I zoom in for you, Jess?
Sure. Go ahead.
Yeah, if we could get some screen grabs of the sediment, that'd be great.
This is sample 039, is that correct?
Yes, NA124-039.
Copy.
Is that all right for the shallowness there, the slurp?
Yeah, it's perfect. It's like vacuuming on Sunday.
Got a lot to vacuum up.
There's all the billions of microbes we just can't see. That's what we're...
See, this is nice because this is a very different kind of sediment than I think we were seeing in the push cores at the base of the escarpment yesterday.
All right, Greg, is that all right there?
It looks, it's a little hard to tell how much is settling in that bin. I feel like
we can turn it off.
Not that much in there.
Yeah, we can turn it off and just have a look.
Is this one of the coarse or the fine?
This is one of the coarse.
Yeah, this sediment might go through.
Yeah, so.
You want some more?
Yeah.
Roger that. All right, Jake, go ahead.
Roger.
I'm wondering if we should have gone for the fine mesh. Not exact sure of the pore size on that mesh, so we may be losing a lot of sediment through that coarse mesh.
All right, go ahead and try zeroing the selection again and see if we get anything else in there, Jake.
Well, that was a good test of this, Jessica. So I'm happy with that. And if we try to do it again, maybe we'll think about trying with a fine mesh. But this looks like a pretty good test of a new way for us to collect sediment. So thank you.
Of course. All right, so you're happy for us to stow?
Yeah, so Greg, I think you want to do the colonization experiment deployments first, or
All
we
right,
want to
cool,
move up a little first before
Yeah,
we deploy
we need to
them?
keep
Yeah.
moving to
Okay, so
deploy
we
them.
want to,
We want to get up into some lower oxygen before we deploy the experiments.
so given how long it took to deploy the last ones, maybe we should mosey uphill a little bit. And
Roger that, continue on to point N.
Jake, go ahead and line it up on the flush there. Thank you.
I'm going to let you get ahead there, Jess. You're kind of directly under and a little behind.
Raj, yeah, I'll hop ahead. And then Jake, we'll have to sort out Mongo when it's like our jaws open and
Yeah, okay.
pinching the ground there.
Okay.
Go ahead and close it up there.
The general direction of point N will be bearing 100.
Raj, 100. Sorry, Jake.
I can do it bubble too.
Yeah, that'd be great.
That looks pretty good there.
Thanks, Jake.
Yep.
Do you mind looking back at the toolbox real quick? Okay. Thank
Did
you.
he tell you about the ballast rock?
No.
I don't know if that's meant to be a sample. We can check with the back row, but I know it's at least for ballast.
Roger that.
Data, that rock on the front porch, do we have that logged as a sample, or is that just for our ballasting?
I believe we had logged that as a sample. We right now have its location as in porch.
Okay.
But it's a really nice sample. We really liked, I think Kira really liked the look of that rock, so we'd love to possibly keep it. So if we can keep it right there for now.
Roger.
Thank you.
Turning to 100.
Roger that.
so as we cruise up this slope, we're going to keep our eye out for a nice location with some invertebrates to collect and also a couple of rocks at the same time, hopefully. So some of the things we're looking for are sponges, and you're seeing some here. One of them we've collected already. Some of them are a type of sponge known as a glass sponge, which can be a little less interesting for us at times for our interest in natural products.
All right, Jess, I'm going to move at point 2.
Roger that. Go ahead.
The
Bridge now.
glass sponges tend not to be very well known for making these types of molecules that we're interested in.
50 meter step, bearing 100, speed 0.2.
But everything here is very new, so we're exploring.
It's quite a slope we got here.
Right.
Really nice rocks.
Is that a crinoid, Greg?
Yeah, could we zoom in on the flowery thing on the right of the field, please?
Yeah, sure thing. All right, Al, go ahead. A little wider, please. That's good.
Yeah, this is a feather star, a crinoid, one of the echinoderm groups. And it's basically, think of a sea star lying on its back. And this kind is a filter feeder. It's got a lot of little things called cirri to hold onto the rock, which are underneath. We can't really see those. It has 10 arms, and we can move on. Thank you.
Raj, full wide, please.
What I'm hoping we'll see today is not, feather stars are very good, but they have a group of relatives called sea lilies, which are on a stalk. And there's a particularly beautiful yellow form that's on this seamount that we've had collected for us before, but I never really got great video of it, so it would be nice to see another one.
So Greg, we have a question in the chat asking why there seems to be so little epifauna on the rocks at this site compared to where we dove yesterday. I don't know if you have any thoughts on that.
We're on the western side of this seamount. We're at 1,000 meters, and we're starting to see declining oxygen. I can only think, on the little information we have, is that there might not be very much nutrients or flow of current right where we're at now. We're hoping when we get to a steeper area of slope, we'll see more flow and more filter feeders. We're seeing a lot of this Faria glass sponge, but that's about it so far. We didn't see any crinoids yesterday, and they're filter feeders, and we have just seen one. This is quite a different water mass. We're much further west than we were yesterday. But this is a very big seamount, and we're on one little part of it, so really could vary quite a lot. We were coming at a different angle up the slope at the Patton Ridge yesterday where we were.
Roger that. Thanks, Al.
Yeah.
Do we know how many rocks were collected in the first watch?
We only collected 2 rock samples. One of them went into a box, and one of them is right now riding on the porch.
Okay.
So we're on the western flank of the San Juan Seamount. It is an extinct volcano. It was an island once off the California coast, but it's now fully submerged. And so what we really are going over is old lava fields, but we're looking for other kinds of rock or rock that has accumulated these ferromanganese crusts or phosphorite that are potentially targets for mining in the future. So we're interested in documenting their occurrence and then the animals and microbes that live in association with them.
And you've heard some discussion about biopharmaceutical potential already on this dive, and one of the reasons for exploring this is to just understand what sort of biodiversity exists in these habitats that may be linked to the production of molecules that we call natural products that may have medicinal value.
Jonathan, zoom in, please.
Wow,
Skeleton's
that's
cool.
really cool.
It's a dead sponge there, and what you're seeing is the lattice of glass fibers that is usually a bit obscured by the living cells of the sponge.
Jonathan, go a little tighter, please.
See that beautiful rods of glass. So these sponges are amazing in their ability to secrete silica.
A little wider, please. Pretty cool.
In a very regular pattern, too. It's remarkable.
All right, come wide, Jonathan. Thank you.
The lattice is so perfect. Processing through our samples earlier today, I got a glass spicule in my finger and definitely didn't feel too good.
Wow.
Didn't tell you they're venomous, Kyla, sorry.
Just kidding.
I really hope you're kidding. Okay, cool.
I am kidding.
So still below 1,000 meters.
I'll call in another step. Are you comfortable with this speed, or would you like to go faster?
This is fine. Thank you, Rennie.
Our goal today is to get up to about 500 meters from our starting depth below 1,000 meters, but we were delayed with the launch because of it was a bit of a swell running and wind, so the ship was rolling a bit too much to deploy the ROVs, so we had to wait. And then we have some colonization experiments, bone and wood and rock, that were deployed on arrival at the bottom. We have another set on board with us to deploy at the end, or near the end, once we get up a bit shallower. But we lost quite a bit of time early on, so we're trying to get up at
There's a big rock.
shallower water to see if we see a big assemblage of sponges and corals that we can sample. We'll also be looking for some more interesting rocks to sample. We're also interested in push coring into sediments, but right now there doesn't look like there'll be any chance of that.
Somebody in the chat just asked what bone type we're deploying. We have cow and pig and chicken.
Hi, Crispin. Yeah. I try these days to put out little pieces of bone for, there's always a tension between how long the bone is out and when we can recover it. And we know, well, we're planning to be back here around this time next year. So I've selected some fairly small bones with the hope that they'll last. And then they're relatively easy to dissect out the bone-eating worms that we're after, and there's also some other kinds of bone-associated animals we hope that will come to this little collection. And we'll be putting out 4 different bone cages in 2 different places at 2 different depths.
We do tend to see a fair bit of those yellow stalked crinoids at deeper volcanic terrains. But I don't recall having seen many in the Channel Islands this year on our dives, and they've mostly been pretty shallow.
At about this kind of depth, or?
What depth are we at here, 1,000? Yeah, well, we didn't have too many dives this deep in the Channel Islands, so I don't know that our data points from this year, and as far as stuff in the prior years, well, they all kind of blend together a bit.
Yeah. That looks like a big shrimp.
Well, we're seeing some great sites to collect rocks, but maybe not so great so far for some of the inverts we're looking for.
Just let us know if you want to pick one up on the way.
And if you're you're wondering why we're interested in sediments, it's because we're interested in the bacteria that live in ocean sediments.
That's a nice big rock.
Yeah. This bacteria, I think, as most people recognize, make lots of these natural products, including many important antibiotics and medicines that almost all of us have taken at some point in our life. And so people have not studied the microbes that live in the ocean for this potential. So sediments are incredibly complex and diverse in terms of the bacteria that live there. So we prefer to collect cores where we can actually section the cores with depth and see how the communities change in those vertical profiles. But when you can't core, you're trying to use the suction. It's another method of collecting sediments.
I haven't checked since watch change.
Seems like we're not getting that much change in depth. Is that correct?
We've actually been going pretty steadily upslope.
It looks pretty slopey.
Yeah, so a few minutes ago we were about 30 meters deeper.
That sample we took was at 1077.
Okay, 50 meter.
You can see it on sonar too, the kind of the rise of the slope there in front of us.
Do you mind if I do a quick zoom in on Herc?
Sure, for focusing.
Just for beauty.
That red shrimp
on Argus. Okay.
Of Herc.
The left there.
On Herc, yeah, sorry about that. Look at that. Whoop, and zoom out.
Gonna continue that move.
Roger that.
Bridge nav. One more step, 50 meters, 100. Thank you.
Lots of different rocks. Nice view from Argus looking at the terrain that we're crossing up.
It's remarkable.
Would you mind on the DVL there, please?
Ah, sorry.
All good.
I don't like the auto center feature. Gives me less control.
Ooh, that yellow.
I'll try it.
That looks like a sponge.
That is indeed.
That is indeed a sponge. You know, a quick snap zoom, partial there, Jonathan. That's good. We'll get them both in view.
Looks
Very
like a
cool.
dead
Thank you.
Ferrea.
Tortilla
Great
sponge.
screen grab. Thank you.
Yep. Full wide, Jonathan.
A little bit different than what we've been seeing.
It's that really spiky thing.
Ooh.
Ooh,
Oh.
yeah, coming up.
Yeah.
Urchin or
Subsea
Ooh.
porcupine.
Like a dandelion.
Okay,
Well,
Jonathan, you want to do
spinier.
a snap zoom there?
my guess is urchin.
Oh, yeah.
Wow.
Just very long spines.
Wow, you spiky.
Very cool.
Super cool.
Fitting well there in the rocks. You don't want to get close to those.
A little wide there, Jonathan.
I think we'll have to go.
Yeah.
Got a wall approaching.
All right, full wide.
Thanks for that zoom.
Yeah.
Some
It looks like,
vertical.
yeah, pretty vertical there.
See that Crispin just sent in the science chat. Probably
Kinda.
just saw a little small serpulib tube too.
Oh.
Probably when we were zooming in there before.
Come up a little faster there, Jake. Thanks.
There's that wall we were talking about.
Yeah.
You want me to hold up here? Give it a
Yeah,
pause?
let's give it a pause there real quick.
Give it a pause.
Something.
Seems to be, Argus
Very
is
cool.
cresting it.
Yeah, it looks like we just passed over a crinoid. Steve just ID'd that for us. That was that stalked star, feathery
I think
looking
we'll
thing.
clear it.
Yeah. Could be all right.
Ooh, that's a cliff right there.
Yeah.
Look at that. It's remarkable how rapidly that changed.
Yeah.
Whoa. Ooh, another one of those spiny sea urchins. One of those yellow sponges.
Yeah, Argus is getting clear of it. It's just this bit here.
Nice.
Now we're above 1,000 meters.
This would be some great rock.
Yeah, it's
Yeah,
very
if you wanna
chunky
All of
rock.
this rock, yeah. Kind of stacked together.
It's all stuck in there.
Like a brick
Wow.
house.
Kira's probably downstairs.
Ah, so these sponges, Steve says, these are still the same Ferrea sponges in this area. And that crinode we passed over definitely looked like a comatulid crinoid.
Some glossiness on this. Manganese or something.
Looks like something was rubbing on that.
Crusting. Wow.
That's cool.
Super spectacular.
Wow.
Steep wall.
Yeah.
Going in the notes.
Wow. Did it level out? No, we're still going up.
Still going up, but it's
Still
not
going
as
up.
steep as
Not
that.
as steep as that really big one we just went over. Okay.
Yeah, it goes up steeper kind of to our right here.
Yeah.
Kind of see it that way.
Larger rock faces there.
Yeah. Very interesting.
We've dropped in oxygen saturation as we'd expect as we're kind of climbing up in elevation here.
Must have lost some beams. I'm going to do a DVL reset on the fly there.
Roger that.
Crispin has commented in the science chat saying that we're going over a lot of this columnar jointed basalt, which is pretty nice. And it looks like this is lava flow with columnar jointing, like in Devil's Causeway in Ireland. It's pretty spectacular to see that under the sea too.
Yeah. Starting to look pretty organized, like especially that wall back there. The way it was fracturing was
right.
definitely.
Kind of the joints.
We are getting towards the top of this slope here, so I don't know.
It's possible at the top we'll see a lot of fauna, so maybe we'll be prepared to stop.
Roger that. If you take a look at our map here, we went approximately through point N, but just kind of kept going directly upslope. Do we need to make it to point O, or should we kind of continue on to P?
That's fine. The new track is fine.
Sure. O is just
There's no need to go to O.
Yeah, a little bit offset by 20, 30 meters. So. Figured it wasn't too big of a deal.
Yeah, you could just track to P. It's fine.
Copy that.
Greg, are we getting in the O2 range you're looking for, or is it still a little too high?
It's still pretty high compared with yesterday. So we'd want to drop it well under 10.
Wow, look at that trim.
We're just watching the oxygen concentration fall as we're going higher. So we started off in relatively high oxygen, still a lot less oxygen than on the surface. But there is a place or a zone, we call it the oxygen minimum zone, which is all along the California coast. And in many other parts of the world where as plankton and other organisms die from the surface, the bacteria and other organisms, and the decaying of these creatures removes a lot of oxygen from the water. So often oxygen can get very near to zero by about 500 meters. And then with deep ocean circulation currents, the oxygen level rises again for the rest of the deep sea. So it's not really deep water that's got the lowest oxygen in the oceans. It's this sort of band at about 500 to 1,000 meters. So we're now at less than 1,000 meters. We're at 975 meters, and we'll see the oxygen steadily decline for the next few hundred meters. And at some point it'll start to rise again.
Like we're going over more of those paralomus crabs.
Getting steeper again, I'll come up faster.
Is that a sponge there? Yeah.
Shrimp in the wind.
Waiting right there on that rock.
Got another 5 meters left in the move. I'll just hold. We'll pause and catch up a bit.
Roger.
They're paralomus crab. Bright red.
A lot of deep sea animals have evolved to be red because out of the visible light, red is the first frequency to be absorbed in the upper pelagic. So it makes sense that a lot of deep sea animals are red because their predators won't be able to see them. But what I don't understand is why some of these animals are yellow. Is there any sort of adaptation benefit for being yellow?
Good question, Kyla. No great answer. It may be the sponges aren't really being preyed upon. These glass sponges have so many spicules relative to their cells that they might not be worth eating. So then in a sense it doesn't matter what color they are if no one's really going to touch them.
looked like that yellow sponge was a yellow glass sponge, ID'd by Steve.
There's another Solus Solitarian.
Yeah, we've got a beautiful
How did that sample come
filter
out?
feeding sea cucumber there on the lower right. It's
Yep.
going out of the shot now. It extends its tentacles to filter feed.
We collected one of those yesterday, correct?
Yes, very large actually. These are very big ones at about 6 inches long. Normally they're only an inch or two at the most, and they stick onto the rock. They're unlike other sea cucumbers that crawl around. These are sessile. They stick on a rock and just filter feed in the water current. Hopefully we'll see some more as we go up.
So just to add a little bit geology, we've got someone on Science Track, Crispin, who just messaged us saying we're going over this fractured pillow lava blocks here with nice radial fractures.
Mm. Oh, that's a really cool piece of.
Look at that.
That's a cool one, yeah.
Yeah, you can see a bit of the radial pattern in it.
Yeah.
I definitely see a sheen.
Kind of seeing
We
both
could do some
ends.
sloping and sampling here potentially. You want to?
Definitely, yeah, I think.
Yeah, we're held up here, so anywhere.
I don't see a lot of sponge action for you.
Very cool rock.
Yeah.
It's definitely flattening out above Hercules, so.
Yeah.
Some of the yellow sponges.
Where the lasers are, there's some sponge, yellow.
Yeah, there, would you like to look at these?
Yeah, can we have a zoom in on that, please?
Sure, let me get a little closer. Right in the crevices there. Okay, Jonathan, go ahead. Oh, they're corals.
Ooh.
Oh my goodness.
Those are cool.
There's a
Very cool.
cup coral in there, looks like.
All right, want to get some of those, Paul?
Steve says
these are
bryozoans.
the yellow or bryozoan.
Oh, that's an interesting group for active compounds.
Yeah.
So.
Yeah. That bright yellow color.
Yeah, let's.
Can you guys get in there?
We can try, for sure.
I think slope might be the best option.
It depends on how large they are. They look
They're
relatively
soft
small.
or hard.
Yeah, and how soft they are.
They're gonna be
Are they
brittle.
rigid?
They'll be brittle.
Yeah.
Come on there, Jonathan. Also looks like there's...
Patch to the right. Doesn't have a cup coral with it. It's just off out of frame.
Are you full wide on her there, Jonathan? Thank you. Oh yeah, that's great.
Yeah, sorry about that.
No, you're fine.
I think there might be one tucked up there.
Oh yeah, it's a great eye next to those sponges there.
That is.
It's so hard to get perspective here. Like how close?
Let's go right
There's a
here.
whole bunch of them.
There are.
Nice, Jeff. That's good.
Eagle has landed.
All right, Rennie, do your magic.
Stick lock.
Raj. All right. Well, we can get a good picture of it here. Jonathan, go ahead and push on in.
Fantastic.
Nice.
Ooh.
Nice trick.
Wonderful.
If this works, we're gonna want to try to slurp a couple of these guys.
Is that a limpet?
Yep, nice big limpet too. That would be good to get.
Somehow thought those were only on rocks on the shore.
So we want to